Hello and hi everyone, my name is Nur Fatiha binti Muhammad Nasir and today I'm going to carry out an experiment regarding the rate of photosynthesis. So the objective of this experiment is to determine the effect of light intensity on the rate of photosynthesis. Plants make their own energy source through a process called photosynthesis. During photosynthesis, plant cells use light energy, water, and carbon dioxide as reactant to produce sugar molecule and oxygen. Photosynthesis takes place in the chloroplast within the plant cell. During the first part of photosynthesis, the light-dependent reaction, chlorophyll pigment absorb light energy to produce an ADPH and ATP, which are two types of energy carrier molecules. At the same time, water is split into oxygen and protons. The next stage is light-independent or dark reaction. In this step, the two energy carrier molecules and ADPH and ATP are utilized in a series of chemical reactions called the Calvin cycle. In the Calvin cycle, the plants take carbon dioxide from the air and use it to ultimately make sugars such as glucose and sucrose. There are many factors that can limit the rate of photosynthesis, which are light intensity, carbon dioxide concentration, and also temperature. The hypothesis for this experiment is when the light intensity increases, the rate of photosynthesis will increase. First, we need plant, soda bicarbonate, water, cup, syringe, ruler, straw, table lamp, paper and pen, mass scale, and timer. First, I'm going to make 10 leaf discs by making a hole using this straw. Then, put all the 10 leaf discs into the syringe and replace the plunger back. Then we mix 0.5 gram of sodium bicarbonate with 100 ml of water to make a sodium bicarbonate solution. And draw the solution 7 ml into the syringe. As you can see, all the leaf discs are floating in the syringe, but we need to make the leaf sink down the syringe. So we need to do a slight vacuum on it by putting thumb on top of the syringe and pull and release the plunger gently. Now we can see all the leaf discs are sink at the bottom of the syringe. Then we pour the leaf discs into the cup. Next, we place the light source 3 cm from the beaker and observe the number of leaf discs floated in every minute. After 4 minutes, we can see that all the leaf discs are floating in the solution. Then repeat all the procedure, but now place the light source 10 cm from the beaker and observe the number of floating leaf discs in every minute. Next, we try to put the beaker into the dark cupboard in 5 minutes to see what happened to the leaf discs. After 5 minutes, we can see that all the leaf discs still sink at the bottom of the cup. So these are the results obtained after carrying out this experiment. When we plot the graph of number of leaf discs against time, we gain a directly proportional graph where when the time increases, the number of leaf discs floated in the solution will also increase. The scientific name of the plant that I used in this experiment is Epomia aquatica, or it is commonly called as water spinach. In the leaf disc assay, all the components necessary for photosynthesis are present. The light source provides light energy, the solution provides water, and the sodium bicarbonate provides dissolved carbon dioxide. After undergoing this experiment, I found that my data supported my hypothesis. When the light source is placed at a distance of 3 cm from the beaker, there are 3 leaf discs float in 1 minute, and in average, it takes 4 minutes for all the leaf discs to float in the solution. It shows that the rate of photosynthesis occurs faster. But when the source of light is placed 10 cm afar from the beaker, there are only 1 leaf disc float in 1 minute. In average, it takes more than 5 minutes for all the leaf discs to float in the solution. So it shows that the rate of photosynthesis occurs slowly. However, when the beaker is put into the dark cupboard and let for 5 minutes, it was found that all the leaf discs are sink at the bottom of the beaker. Alright, from the procedure that have been carried out, the suction in the syringe does help the leaf to sink because leaves have air in the space between cells, which help them to collect carbon dioxide gas from the environment to use in photosynthesis. So when we apply gentle vacuum to the leaf disc in solution, the air is forced out and replaced with solution, causing the leaf to sink. Okay, the second one is, if we conduct the experiment without baking soda, there will no leaves arrive because this bicarbonate solution is taken out by the plant to provide a source of carbon dioxide that is then used by the plant to produce oxygen. This oxygen increases the buoyancy of the leaf disc. And the third, why do the leaf discs in the baking soda solution begin to float? Alright, the leaf discs take up carbon dioxide from the baking soda solution and sink to the bottom of the cup. When exposed to light, the discs use carbon dioxide and water to produce oxygen and glucose. Oxygen released from the leaf form a tiny bubbles that cause the leaf to float. And the fourth one is what happened to the leaf discs in the dark. So when the leaf discs are putting in the dark, they will eventually sink because without light energy, no photosynthesis will occur, no oxygen will produce. However, respiration continue in the dark, so the disc will use to accumulate oxygen gas and they will also produce carbon dioxide gas during the respiration. As a conclusion, this study indicates that the increase in light intensity, the higher the rate of photosynthesis will occur. It is proven when the light source is put near the beaker, all the leaf discs are floating faster compared to when the light source is placed far from the beaker. So, it shows the increase in rate of photosynthesis.